This will be our last podcast for epithelium, and it will be fairly brief. Now, we have, in our previous two podcasts, we've looked at epithelial cell polarity, and we've considered concepts on the apical domain of epithelium. And just to remind you that the apical domain is always directed to the exterior surface or the luminal surface of an enclosed cavity or tube. We've looked at junctional complexes on the lateral domain of epithelium, and we've emphasized that these junctional complexes are important to hold adjacent cells together, and they play a role, of course, in cell-to-cell communication. In this last podcast, we want to talk a little more about the basal domain of epithelium and remind you that all epithelium rests on a basement membrane, and this functions to anchor the epithelial cells to the underlying connective tissue. With regard to the basal domain, we can talk about cell to extracellular matrix junctions that anchor epithelial cells to their extracellular matrix, and we've already mentioned focal adhesions and hemidesmosomes. Now, oftentimes you'll see basal infoldings of the plasma membrane to increase surface area, and this will facilitate morphological interactions between adjacent cells and extracellular matrix proteins. Sometimes you'll also see membrane infoldings to increase surface area depending on the physiologic function of the epithelium. So here, for example, in this electron micrograph, we show membrane infoldings in renal tubules, and we can see interdigitated mitochondria in these membrane infoldings, and this is very characteristic of cells that are involved in active transport, which of course occurs in some of the renal tubules. The mitochondria provide the ATP energy for active transport. Now, the basement membrane is a specialized structure. It's located next to the basal domain of epithelial cells, and it involves the underlying connective tissue stroma. Now, the basal lamina is synthesized by epithelial cells, and the reticular lamina is synthesized by connective tissue cells. Taken together, the basement membrane consists of a basal lamina and a reticular lamina. We're going to talk about two components of the basal lamina, a lamina lucida and a lamina densa. The basal lamina, as we said, is synthesized by epithelial cells. We're going to suggest to you that the lamina lucida may well be a preparation artifact, although it is talked about in histology textbooks as a distinct structure. The major protein in the basal lamina is collagen type 4. There are other collagens involved in the basal lamina. In addition to other collagens, there are laminins. There's proteins like intactins, proteoglycans, and fibronectin. But for the purposes of this course, the major protein associated with basal lamina is collagen type 4. The basal lamina, as we said, is synthesized by epithelial cells. The major protein in the reticular lamina is primarily collagen type 3, and as we said, the reticular lamina is synthesized by cells of connective tissue. Now, what are the functions of the basal lamina? First, the basal lamina supports epithelium, and it separates epithelium from the underlying connective tissue. The basal lamina can induce differentiation of epithelium. It will also contain ligands for cell surface receptors in the epithelial cells. These receptors, when they bind their ligand, can initiate signaling cascades, which, as you might imagine from your studies in biochemistry, can establish second messenger functions and can turn on transcription factors to affect gene expression. Basal lamina can influence metabolic activities of the epithelium. The basal lamina, especially in the glomerulus of the kidney, performs a very important function as a selective filtration barrier. The basal lamina also enhances wound healing and epithelial repair. It serves as a scaffold essentially for repair, and the basal lamina of epithelium can enhance nerve and muscle regeneration in injured organs. This is a simple-minded cartoon, and it makes the point that the basal lamina is a product of both epithelial cells and connective tissue cells. So here we show some epithelial cells in diagrammatic fashion, and here some connective tissue cells. The basement membrane consists of the basal lamina, which is synthesized by the epithelial cells, and the reticular lamina, which is synthesized by connective tissue cells. And by the way, the most common connective tissue cells would be fibroblasts and smooth muscle cells. As we've suggested, the lamina lucida may be a preparation artifact. It appears in tissues when the tissues are fixed with chemical fixation, but if one does more modern low temperature, high pressure freezing techniques, you don't see a lamina lucida. Components of the basal lamina. As we said, collagen type 4 is a major component. It is unique to the basal lamina. It's found in the lamina densa. Collagen type 4 provides the structural framework for the basal lamina. It is heavily glycosylated. Collagen 4 forms relatively small fibrils that do not form the large fibers that you studied in biochemistry. Collagen 4 is highly flexible. The C-terminal ends can dimerize, as suggested in this cartoon, and the N-terminal ends might form networks with each other. Heparan sulfate is another component of basal lamina. Heparan sulfate basically are sulfated glycosaminoglycans. These sulfated glycosaminoglycans provide a fairly high negative charge to the basal lamina. There are other proteins like entactins that form almost like a glue type structure. And then finally perlicans, which are larger components that are combinations of heparan sulfate and some of the various entactins or protein glues. Another fairly significant component of the basal lamina is laminin, and laminin is a large cross-shaped protein. It is on the order of 1,000,000 Daltons, and laminins bind various components of the basal lamina to each other and to epithelial cells. Laminin is actually a protein consisting of 3 different protein chains: an alpha chain, a beta chain, and a gamma chain, and that's shown here in this kind of cross configuration. And then it shows there are regions in the laminin molecule that bind to cells, there are regions in the laminin molecules that bind to collagen, and there are regions in the laminin molecule that bind to heparan sulfate. So the diagram on the bottom suggests that this laminin, not only is it binding to cells, but it's binding to a variety of some of the extracellular matrix proteins. Here is a more complicated cartoon of this, with the diagram on the top showing the epithelial cells, and so the basal lamina highlighted here would be shown here in the diagram. So if you imagined flipping these epithelial cells upside down, here is the lipid bilayer of the epithelial cells. Here are some of the integrin proteins that we talked about in our last podcast, and then a variety of these extracellular matrix proteins, and you can get a sense of the fairly substantial meshwork that is forming in part of this basal lamina. And note that integrins, as transmembrane proteins in the epithelial cells, have receptors that bind onto some of these proteins in the extracellular matrix. Now, this interwoven network of proteins in the basal lamina determines its function. So you'll have structural attachment of epithelium to the connective tissue. There'll be compartmentalization such that epithelia and also nerve and muscle are separated from connective tissue. As we said, the basal lamina serves as a filtration barrier in the renal corpuscle. It allows movement of substances to and from epithelium, and that filtration is in part regulated by the basal lamina. The basal lamina can serve as a tissue scaffold guide for cell migration during morphogenesis and regeneration, and it can also serve as an important barrier to invasion of cells during tumor metastasis. The basal lamina plays a role in cell signaling and regulation during morphogenesis. It plays the same role in fetal development, in wound healing, in proliferation. It's involved in motility of cells, and because it has receptors in the membrane that will recognize proteins in the basal lamina, it can play a role in gene expression and also in apoptosis or cell death. Something we're going to talk about in more detail as we go along in the course. Cells can be polarized, and so epithelial cells can be polarized both structurally and functionally, and as a result, epithelial cells may have a certain amount of asymmetry to them. So we can talk about asymmetry of cell surfaces. We've already talked in our previous podcasts about apical, lateral, and basal domains in epithelial cells. The apical domain may have cilia on it. It may have microvilli on it or stereocilia. There could be a glycocalyx, which is oftentimes PAS positive, and this can be associated with the apical domain of epithelial cells. The lateral domain of epithelial cells will contain junctions that bind cell to cell. These junctions can hold the cells together, and these junctions can also play a role in cell-to-cell communication. The basal surface of epithelial cells will have junctions that bind the epithelial cells to the extracellular matrix, and the membrane in the basal areas may be thrown into folds to increase membrane surface area. There can be distinct plasma membrane domains in epithelial cells, so the apical domain may have various ion permeabilities. We looked, for example, at the sodium glucose symporter. The basolateral domain may have glucose carriers, especially in epithelial cells. There'll oftentimes be sodium potassium ATPase molecules at the basolateral domain of epithelial cells, and this will be important in maintaining the ionic differences across cell membranes. And epithelial cells can be polarized with respect to their organelles. They can be polarized in the biosynthetic or exocytosis pathway, so there'll be an array of ER, Golgi, and secretory vesicles that will move towards the plasma membrane. Epithelial cells may also be polarized in the endocytic pathway, so you could absorb lipids, you can have diffusion, there can be resynthesis, formation of chylomicrons, and this is all things you may have studied in biochemistry and that we'll touch upon as we go through the course. This cartoon gives an example of polarization of epithelial cells with respect to plasma membrane domains. So you may be bringing material into the epithelial cells from a lumen, and these materials may transport across the epithelial cells and may be exocytosed at the basal side of the epithelial cells. As we suggest, there are some junctional complexes in epithelial cells. We've talked about the sodium glucose symporter. We will talk about endocytic and exocytic vessel formation. We've talked about glucose carriers, and we've mentioned, for example, the sodium potassium ATPase, and there are sodium potassium antiporters that can be found in the basal domain of epithelial cells. We will talk about cell polarization with respect to organelles, and we will talk about cell polarization with respect to vesicle flow in cells.